Would you like to speak as well?
Yes, my students are presenting. Well, I mean, what I can go to. Okay, guys. How are we doing? I saw half. I was just running back to my car to look for my keys. That seems to be MIA, so I'm a little bit like. And half the class saw me. I was like, and I was like, we're okay. I'm hoping my keys aren't in my front door. That's always not a good place to find them when you get home. And you live in Baltimore City. So usually, well, okay. Okay, pick up exams and recitations if you haven't. Gene expression topics need to be finalized this week. It's looking good so far for Monday's recitation. Remember, this is a proposal, not a report. You are forming a hypothesis based on the data and proposing a way to study it. So starting with reading everything about CMV and trying to shoehorn the data into something isn't the way to do it. You want to look at the data and ask yourself, does this make sense? Is there anything weird about this? There are certain things that are obvious. If you're infected with a virus, you should be mounting an immune response, right? So if for some reason immune-related genes are downregulated, that's interesting. But if it's like, oh, this is very exciting because it's highly upregulated and in its immune response, well, tell me why it's exciting. Don't we expect that to happen? Why should you study it? What's your question? So if you can't develop a question, then you might need a little help, and that's why I'm here. And if you don't need help and it's just that there's no question, then you need to drop that topic. Exam 3, coming right up. Okay, so far we talked about the major concepts of gene expression and what it is, why it is, all that good stuff. Similarities and differences, DNA versus RNA, DNA polymerase versus RNA polymerase. We talked about transcription in bacteria and eukaryotic cells. Transcription is coupled to RNA processing, right? That's capping, splicing, polydenylation. This is a major concept, make sure you understand how and why and all that. And we're going to be doing that in recitation today. You guys are going to be drawing out, or today and Friday, you guys are going to be drawing out this coupling. So you're going to be showing your RNA coming out of your RNA polymerase and the splicing reaction happening. So the splicing factor is bound to the C-terminal domain that's been phosphorylated and then transferring onto the splice sites or branch point as they're exiting the RNA polymerase and then going through the steps of the splicing reaction. And we started introducing concepts of DNA elements. DNA elements are sequences of DNA that are involved in regulating transcription, so they're going to be bound by transcriptional regulators. That's the next one. We talked about promoters, enhancers, and locus control regions. These are all DNA elements involved in regulation of transcription of a gene. That's where we are. Okay, so this is where we were ending. Okay, so we're talking about what regulatory regions look like and what they do. And so here's another thing to think about. DNA binding proteins don't directly control transcription, but control transcription through the proteins that they recruit. Okay? So when you've got things binding to a promoter, it's recruiting RNA polymerase. And when you've got things binding to enhancers or other regulatory regions, it's controlling transcription also through recruitment of general transcription factors and RNA polymerase, and also recruitment of chromatin modifying enzymes and those kind of things. Right? So you have to also reorganize your chromatin. Okay? So this is showing recruitment of co-activators and co-repressors. So these are things that interact with transcriptional machinery or modified proteins. So you can have an activator that might recruit a co-activator that then might recruit some enzymes that are going to modify the transcriptional machinery, or that's going to recruit transcriptional machinery or modify chromatin. Yes?
I'm sorry. You said the exam is not next week.
Oh, I have no idea. Whatever's on the most recent syllabus. I believe I said the 29th, week of the 29th? Yeah. So those will be puts out of next week if you're looking at an old version of the syllabus. So always check on Blackboard to make sure you're looking at the most recent for some time. I usually let you know when I'm posting a new version. There's only been one modification. So you can have co-activators and you can have co-repressors, and that's where you have an activator recruiting a co-activator, which recruits other things that are going to be involved in activation or repression. And the other concept to think about is that regulatory proteins, their activity can be regulated through modification. So whenever you think about anything, there's a level of regulation that's going to occur. And a very common way of regulating something is by modifying the protein post-translationally, right? You can either have a regulatory protein modified by some kind of allosteric effector like cyclic AMP, which is going to bind and change its conformation. Or you can also have covalent modifications such as phosphorylation. They're going to change the shape of the protein and change its ability to bind to the DNA in some way. And we're going to go through multiple examples of all of these. These are just major concepts to understand. Okay? Makes sense, right? If you've got a protein that needs to bind to DNA and you phosphorylate it, maybe it won't bind to DNA anymore. It's not in the right shape to bind to the DNA, it won't have the DNA binding site anymore. Okay. Activation or repression of gene expression can occur through multiple things, but one of the things is through recruitment of chromatin modifying enzymes. So all that stuff we talked about in the first few weeks of class with all the methylation and acetylation, all that stuff, don't forget your chromatin needs to be modified in such a way that it's available, that the DNA is available for transcription, right? So some of the proteins or complexes that are going to be recruited by activators might be chromatin modifying, okay, or chromatin remodeling complexes. And so this is just showing all the different types of modifications that can be happening on one gene. Lots of modifications. Remember, we learned about all this when we looked at how you can have lots of histones being modified in many different parts of the tail, right? Yes, no? Okay, so how do transcriptional regulators bind DNA? Have you ever thought about it? They just bind. DNA all looks the same. So if you think about a protein-protein interaction, proteins are very different in structure based on their sequence, right? The amino acids and the way they make up a protein is going to determine their structure. And that's going to create certain binding sites, right? When you have proteins binding to DNA, wait, DNA is a double helix. How does a protein know to bind here and not here, right? Have you ever thought about this? Maybe a little bit. I'm getting like a good. Okay, so how do you guys think transcriptional regulators might bind to DNA? What aspects, what properties of the DNA, what structures in the DNA might be important for binding to transcriptional regulators? Now, I'm going to give you a hint. Transcriptional regulators are sequence specific. They bind to specific DNA sequences. Yes?
The major because it has a higher affinity for regular base because of the additional base that it can.
So the major group, so you're bringing up the concept of the major group and the minor group. So remember when you guys learned about DNA the first time, someone said something about the major group and the minor group and you said, right? And you didn't really think about what the purpose was, you're just like, okay. So we're gonna look at that a little bit more deeply, but basically, inside the major group of the DNA, you have access to a lot of hydrogens for H bonding that are specific to the different bases. So when you have a specific sequence, it's going to kind of make a different binding site. And we're going to look at that. Yeah? DNA folding. DNA tends to be packed into chromatin, and that's about it. It's not like RNA where it creates the secondary structure. Yeah, so the comment was, we have an instance where DNA is bent and a site is exposed. And yes, actually, what you're going to see shortly is that the sequence of the DNA does affect its structure. It actually does. And certain sequences tend to have a bit more of a bend, and then you will have a greater binding site exposed. Yeah. Okay. Good comments. Any other comments or thoughts? Yeah. Okay. Okay, so here's what we're talking about. The concept here is about the binding of DNA, binding proteins to DNA, because that's what regulates transcription, right? And we need to understand how that happens. So here's DNA, and there's the major groove, which is the big V, and the minor groove, which is the little v, right? You see how this is big and this is little? And on the opposite side, you've got big and little, right? And so just the way that the DNA is, the structure of the DNA creates this major groove and minor groove. And this is where the DNA binding proteins access the bases from at the edge of the helix and mostly within that major groove. Okay? So each and every large V, I call it V because it's like a V from the side, right? Alligator mouth, maybe? I don't know. Each and every one of them has the ability to bind some kind of protein. Whether or not it's going to bind a protein to activate gene expression, blah, blah, blah. That's all sequence and protein specific. And that's where it's just like when you start thinking about proteins binding to each other, where it's dependent on those chemical interactions between different amino acids, right? They all have different biochemical properties, and that's what determines proteins binding to proteins. Well, proteins binding to DNA, it's quite similar in concept, okay? So here's how it can look. So if you're looking straight down, right? So you can see, you're looking straight down, you can see your major groove has this huge amount of space. So here's a GC base pair, right? And you've got this huge space. So this is looking straight down the helix. Yes?
Are those where you would find promoter in the major groups?
Promoter regions are going to have probably a combination, right? Because the major groove and minor groove are every, how many, 10 base pairs or something like that. I have it there. 10 nucleotides per helical turn. So, and some promoter can be really large, so it's going to be a combination of major, minor, you know. But that's going to create a number of binding sites. Okay, let's go back though to this. So if you're looking straight down on the helix, you see here's a GC, here's an AT, and if you look within the major groove, you have access to all these potential H bond donors, right? There's a whole bunch of H bond donors here, and here we have a methyl group, which is hydrophobic, and then you also have a number of H bond acceptors, which are in red. So the donors are blue and the acceptors are red. And you're not opening up the helix, but just you're able to access the bases from the side. And that's how those proteins are able to bind. Okay, are you guys following? Some nodding? Okay. Okay, so if you look directly in, so we were just looking from top down, So now we look straight on. I've got a double helix in front of me. I'm looking right into the major and minor grooves. And you can see, depending upon the sequence, you're going to have different combinations of hydrogen bond acceptors and donors. Right? So in your major groove, it's larger, so there's more to access. And in your minor groove, it's smaller, so there are less opportunities to access. And based on what protein is acting as the transcriptional regulator, there are going to be these conserved DNA binding domains that we're going to talk about. So remember, domains, these are just functional units of a protein. So there are all these DNA binding domains that specifically bind within the major groove under certain conditions. Okay? Yes?
So if both DNA sequences and protein present, and what would you do from binding to DNA?
Tell me, come again?
I'm sorry. If both protein and DNA sequences are present, like what prevents proteins from binding to DNA sequences? You know what I'm saying? It's like they both of them are present.
If both, okay, so if both proteins and DNA sequences are present, so you've got proteins, which are your transcriptional regulators, and DNA sequences, which is your DNA and your promoter. So they're both there. What prevents them from binding? So you're saying how come it's not just binding willy-nilly anywhere?
Where's the regulation?
Where is the regulation? It's specific to the sequence of the protein and to the sequence of the DNA.
If both of them are present, so.
Well, they're both present in every case, right? So you always have DNA binding to protein, right? Whenever you have regulation of transcription, you have to have some kind of transcriptional regulator bind to a DNA element. Okay, and so depending on the sequence of the protein and the structure of the protein, it's going to have structures that are going to bind efficiently to the DNA in the major groove, or not if it's not the right. So okay. So there's a few ways to answer the question. One is just based on can it bind or not? And the other is it there or not? Because remember, in order to bind to DNA, you need to be in the nucleus. And if you think about, are you taking 408? So when you think about signaling, usually the end result of signaling is sending a transcription factor into the nucleus, and then it can bind to DNA, right? If it's not in the nucleus, it can't bind. So there are a number of pathways that function by keeping transcription factors outside, all of them pretty much, by keeping transcription factors outside the nucleus. So that's one thing that needs to happen for activation or repression or whatever needs to happen. You need to actually get into the nucleus and be near the DNA. Then you have to have a structure that wants to bind to whatever pattern. Because here it's showing GACT. What if it was AAAA, right? It would look different. It would have different abilities to interact with proteins because the ability to H bond would be different, right? Did I answer your question?
Yes.
Does anyone understand her question? Do you want to expand? Anyone want to expand? If I'm missing it, yes.
Or if they're just domains on certain tails that also prevent binding proteins.
Are there domains on histone
Or
tails?
constituent defenses, so to speak, to keep proteins from binding to DNA prematurely.
I mean, well, so right, that's another thing. So if your chromatin is organized in a highly condensed way, you also won't be able to access binding sites on the DNA.
Okay, so I have one more question. So if the protein is present inside the nucleus and the right DNA sequence is also present, so then we're going to do
Yeah, so if the protein's in the nucleus and the sequence is available and it's the right one to bind, it should bind. Yeah? Does everyone agree? I think so. I'm saying yes. You had a question?
Yeah, if you're looking at a strand of DNA straight on like you were talking about, and you're looking at the major groove,
So like right there.
yeah, if you turn it 180 degrees, you're still looking at the major groove.
You're looking at the minor groove then. So if you go back, let's go back. Because every single time, so on this side you'll have the minor groove, and then here you'll have a major groove.
Well, then how do they like bind to goes to the major groove, they're binding to the sides. Like,
You're binding to,
really, like, the opposite side is the minor groove, so it's kind of binding to the minor groove too.
we're gonna look at, so the question is, if you're a group, but if I'm saying it's binding to the major groove, but the opposite side is the minor groove, then how is it just binding to the major groove? I'm not saying that it's just binding to the major groove. I'm saying the initial contacts that are going to secure that interaction are within that major groove. There might be some kind of interaction, but usually DNA binding proteins are not wrapping around the DNA. They're kind of like imagine how a person sits on a horse, flopping down, or they might stick it. Well, so what we're going to do? We're going to go through a bunch of examples, which will hopefully get you to visualize exactly how this is happening. Hopefully, yes.
I think my question is still on the line what she said.
Okay.
How are these transcriptional regulators? Are they just always activating the DNA? They're always just floating around the nucleus and just opening up? Okay.
Right. So this is connecting the concept of signaling and the concept of activation of gene expression in response to something to this whole process. So let's go back to the big picture, which is gene expression is activated only when it's needed for each gene, right? So if you need to make hemoglobin in response to hypoxia, you need to be hypoxic, which means needs to result in activation of a transcription factor that's going to go into the nucleus and bind to certain HIF. Its HIF is one of the major ones. And will go into the nucleus and bind to HIF responsive elements in the DNA. That's how it's regulated. There's no hypoxia, HIF isn't stable, it's actually constantly being degraded. So it's never available to go into the nucleus.
Don't you need to like activate a gene to make that one protein to activate the other one?
Absolutely, yes. So it's like, it's a cycle. So what's going to happen is you're going to have, in that example, you're going to have low oxygen, which is going to cause hydroxylation of HIF. So all the oxygen is going to modify HIF, so it can't be degraded anymore. Then HIF's going to go into the nucleus, activate a ton of gene expression. One might be making hemoglobin, one's making more HIF. So you see how it goes, and it all starts to kind of feed back on itself. Once whatever happens to that oxygen levels, hydroxylation actually results in degradation of HIF. So I did it off, but whatever. So the oxygen is sensed by the transcription factor. And then it either is degraded or it's not. And under low oxygen, it goes in and activates gene expression of all these different genes for making hemoglobin, and genes for making new red blood cells, and genes for making blood vessels, growth factors, all these things, and HIF.
So they all, every cycle really starts with a response to the environment.
Absolutely. So low blood sugar, that's going to activate another pathway. High blood sugar, activate another pathway. Any bacterial infection, activate a million pathways. Viral infection, you've all seen, activates a whole lot of stuff, right? Yep. But always remember, you've got something going on outside of the cell or within, but there's something being sensed that then activates the transcription factors and then activates gene expression. Okay. Any other questions? That was a good one because it's good every now and then to like back out of the major groove and just think of the big picture. Okay. Okay. So we've looked at all this. Okay. So then there's this question: is the double helix uniform in structure? So you would think yes, right? Because this is what you always see. But in actuality, this is an electron micrograph of 200 base pair fragments of DNA. I think it's right out of our book. And each one has a different shape, right? And that's because the sequence of the DNA actually does affect the structure of the DNA. These are just 200 base pair fragments. So normally it's not going to exist that way. It's going to exist as part of a whole chromosome. But there are these small differences in structure, and also the sequence is going to affect the bendability of the DNA. So for example, some sequences, I think more, I forget which ones, probably more AT-rich sequences, are are able to distort the DNA more easily. And so some DNA binding proteins will bind, and look, you see it, they're sticking in. See how it's sticking into major groove? You see that, everyone? And then it'll actually distort the shape of the DNA. Okay? Okay, so now let's look at what a DNA protein interaction looks like. So this is the DNA, and this big glob is the protein bound to it. Okay? And so you can start to see how it works. You know, so here's a major groove, here's a minor groove. Depending on what side you're looking at, there's the major groove and minor groove, they all seem to shift, right? Because it's a double helix. But there are certain biochemical properties of the protein that are going to encourage interaction, and then there'll also be a bit of electropositivity on the surface of the protein, because remember, the backbone of DNA is negatively charged, so that's also going to help to bind the protein. So there's that kind of general electronegative surface, and then you're going to have the more specific DNA binding domains that are going to stick in to the major group. Okay, so this you can start to see it here. Okay, so I just said, we just discussed it briefly. How do protein DNA interactions compare to protein-protein interactions, and how do they differ? So we kind of talked about it, but who wants to tell me how they're similar? We talked about it, so I'm making it easy on you, but I'm guessing none of one is ordered, so we'll discuss for a minute. Jason? Oh, no, you're busy writing. How do DNA protein interactions compare to protein-protein interactions?
Well, it's probably like influenced by the charges. Negatively charged proteins will bind to positively charged proteins, and negatively charged DNA will bind to positively charged proteins.
Right, so on a very basic conceptual level, it's a lot of charge-based interactions, an ability to hydrogen bond. So a lot of those biochemical interactions determine protein-protein and protein-DNA interactions. So you can think of those as kind of similar. How do they differ? Yes?
Protein DNA interactions are sequence specific, while protein interactions I think is more of a domain. Not domain, but like how they
Protein DNA interactions are sequence specific. Okay, so certain domains, certain DNA binding domains like to bind to certain DNA sequences. And protein interactions do have a lot more of a secondary and tertiary structure, right, that's involved in their interaction. Spilling coffee myself, I'm so excited about DNA. Okay, yep.
Protein interactions being more specific, protein bound to one another, versus DNA will.
So the question is, will protein-protein interactions be more specific and DNA-protein interactions are less specific? That's scary. I hope not. I hope they're highly specific, because we don't want to be upregulating genes when we don't want them by accident. I mean, yes, there's this huge amount of like electronegative, electropositive kind of interaction, but then you also have these very specific interactions within the major group. Yes?
My rebuttal to that would be you're only dealing with 4 base pairs with DNA, whereas protein has a
Yes.
crazy secondary tertiary structure, which needs to be specific.
Yes, you're only dealing with 4 base pairs, absolutely. But at the same time, what it looks like as you look into the major groove can differ insanely based on the order, right? Because you're dealing with this combinatorial nature. So yes, less bases than amino acids, but also don't forget a lot of amino acids have the same biochemical properties. So there are a lot of similarities. similarity.
Is there a difference in the strength of the interactions?
Is there a difference in the strength of the interactions, protein-protein versus protein-DNA? I don't know. I think they're all individual because every protein-protein interaction has its own binding constant, and every protein-DNA interaction can have its own binding constant. Yeah, good question. Okay. How are we doing? Okay. Did anyone have friends in Boston?
Yeah, actually, I'm living with a family friend, and he was running the race. He finished an hour before the explosion.
Thank God he finished an hour before the
Girl
explosion.
at Towson, good friend of mine from high school. She like APTA series.
Like,
And her sister and her mother-in-law also entered, so we might hear something from the Towson.
yeah.
I don't know if we're gonna do something for her, she's a grad student here.
Yes, really not happy. I ran that race 3 times. Crossed that finish line 3 times. Crap. Was my time crap? I was abandoned. I didn't qualify, so it didn't make a difference. I was like in the 4.5 hour mark. Not pretty. Yeah.
So what were this specific?
Okay, let's go back. Sorry, it's on my mind because I've lived there for 7 years. Okay.
So what were the specific instances of difference between protein DNA and protein?
So differences, I'm thinking, are mostly that your DNA is going to vary in shape a little bit, but not nearly how it's going to vary, how proteins vary in shape, right? The DNA structure is a double helix, so it might have a bend to it, but proteins are vastly diverse in shape and have many binding sites. Okay. Yeah.
These sequence-specific like binding sites. How many base pairs are they usually consist of?
Well, I'm going to show you a figure. So the question is, how many base pairs are a sequence-specific binding site? Usually it's just only a handful of base pairs. I'll show you a chart which has a few of them. People make whole careers out of trying to predict transcription factor binding sites, writing algorithms to come up with these consensus sequences. It's pretty amazing. And they're not, some of them are very good, but some are not so good. Okay. There you go. So here's a handful of gene regulatory proteins and the specific sequences that they recognize. And sometimes all you need to do is mutate 1 or 2 bases and you lose that interaction. So they're fairly specific, which you want. You're regulating gene expression. You don't want to be turning things on or turning things off. Just like, oh, today we don't feel like binding. Okay. So gene regulatory proteins are binding to DNA through interactions between amino acid side chains and atoms available for hydrogen bonding in the DNA major groove. And so here's another way of looking at it. You're looking straight down again on your helix. So remember, I'm only showing 1 base pair. That's for simplicity. There would be a lot of them. And we're only showing 1 side chain of an amino acid for simplicity, but normally you'd have 10 to 20 contacts. And so here's asparagine binding here. Okay? So that's what 1 amino acid kind of reaching in and binding in that major group would look like. Questions? Thoughts? Okay, so now we're going to go through a few common DNA binding motifs. So a motif and a domain. So let's talk about that for a minute. So domains are functional units of a protein, bless you, that do something, right? We've learned about this many times. Chromodomains, SH2 domains, a million domains, helicase domains, ATPase domains. So these are all parts of a protein that do something. So when we start talking about DNA binding domains, it's actually more frequently we're talking about DNA binding motifs, and these are just shorter. They're shorter than domains. Domains sometimes are almost like their own secondary structure, whereas a motif could just be a string of amino acids that's important for this interaction. So a motif might be within a domain. So these are DNA binding motifs. So a short stretch of amino acids within a protein that are required for binding to DNA, likely in the major groove. Okay, in a sequence-specific manner. Okay, so here is a common one called the helix-turn helix. The reason it's called the helix-turn helix is because you've got a helix and a turn, then a helix. Right? And so this is how it would sit in the major groove. So there's a few ways of looking at it. So this is from molecular biology of the cell, and actually the helix kind of just lies. Now this isn't one protein, it's just showing the DNA binding motif. Okay? This would be a part of a very large protein, and this part of the helix, this helix-turn helix motif, would stick into the major groove. So you can look at it in a few different ways. Craig, who is the author of the textbook I used last year, showed it in a different way. And so here it's showing multiple different proteins that have the helix-turn helix, right? So this is one, this is CAP, which if you remember, we're learning the left operon, that's involved there. The lambda crow, lambda repressor. Notice there's a lot of structures going on, but only in each of these there's two helix-turn helix motifs, okay? And then when you see how it interacts with the DNA, it kind of sets up like that, where it sticks that helix right into the major groove, okay? You can start to see how that interaction happens. And so you can imagine that this would be a common structure or common motif that would be present in DNA binding proteins because it works, and so it's been conserved. Yeah?
What is the helix describing?
What is the helix describe?
Structure of
The structure of a protein, the secondary structure. Right, because there are alpha helices and beta sheets. Remember learning about all that? You guys learned that a million times and probably said, yeah, proteins do this. But they're functional, right? So when there's a certain sequence of amino acids, they want to interact in such a way that they end up in secondary structure forming a helix. So it's a helix, and then there's a change some here, probably a proline, I don't know, that will then shift it a little, and then you'll have another helix, and that's just sequence space. Yeah. Okay, so that's a very common one, and I like the way Craig, I didn't like much about her textbook, but I did like the way she did this stuff, because it really shows nicely how that helix is kind of sticking right in there and how it's accessing the bases from the side. Okay, so you can imagine you're just seeing a little cylinder, but there are side chains sticking out, right? And they have the ability to H-bond with the DNA. And that's how they're binding. Any questions about this concept? I don't know, it's kind of an amazing moment in my education when I started to really understand how proteins and how things interact at the molecular level. It's kind of cool. I don't know, but maybe that's why I'm a geek, and you're not, or you are. Okay, so here's a homeodomain, and it's a monomeric helix-turn helix, so it's one monomer helix-turn helix, whose binding is stabilized by an N-terminal arm that forms additional contacts with the DNA. So here's your helix-turn helix again, right? So it's showing your blue and your red. There's another helix there too, actually. But it's this helix-turn helix that we care about. And then here's this N-terminal arm. The way it works is you've got your third one sticking into your major groove and making these contacts with your bases from your DNA. And then you've got this N-terminal arm that's not part of the helix-turn helix that's sticking out and making another contact with the DNA. So that's not necessarily specific to the major group. It's just another contact with the DNA. So that's a homeodomain. You do not need to memorize all these, and they're all examples, because you should understand the major concepts with how proteins bind to DNA. Okay, and so this is here how Craig showed it. So it's actually showing it's a helix here. So here they show it as this kind of cylinder. Looks like a toilet paper roll. But here it's actually showing the helix of the protein, helix to terminal helix. And then here's this amino terminal arm that kind of sticks out and makes extra contacts with the DNA over here. Okay? Everyone having fun? I hope so. Okay, zinc finger. This made it onto the quiz. The zinc finger is the most common DNA binding domain in the human genome. Zinc fingers don't always mean it's a DNA binding domain. Zinc fingers are involved in a lot of enzymatic functions too. They're involved in stabilizing all sorts of structures, but it's very common in DNA binding proteins. So that's why we bring it up. The structure, so when we talk about zinc finger, it means that a zinc ion is binding to side chains of amino acids, usually his and cis, and stabilizing the structure of the protein. So on the quiz, I had a question about zinc chelators, and I explained that zinc chelators bind to zinc and they can remove zinc from a protein. And so you can imagine if you pulled the zinc atom out of this protein, it would probably affect the structure. And since that structure is important to binding to DNA, then it would probably affect the ability of the transcriptional regulator to do whatever, do its job. So here's a transcription factor called ZIF268, and so that's all the blue. And you can see it has multiple zinc fingers. So sometimes the transcription factor will have multiple of these domains next to each other, and they're all going to bind to the DNA and make a tight interaction. And the zinc is not binding to the DNA. The zinc is actually coordinated in the middle of the protein by cysteines and histidines and helping to keep the protein in a certain structure. And a lot of protein structures require metal ions, sometimes for enzymatic activity, but with zinc we frequently see it as just stabilizing the structure. Okay? I can't say enough, the zinc is not binding to DNA. The zinc is not binding to DNA. Okay, so if that comes up on an exam, don't tell me the zinc is binding to DNA. Okay. So zinc is coordinated by cis and his, so cysteine and histidine, and it could end up looking like this. If this is your protein sequence, your amino terminal, and your carboxy terminal, right? Your N and your C terminal, so beginning and end. You have a sequence that's shown here just in single letter, and then you'll have histidines and cysteines spaced in such a way that they're going to come together and bind to the zinc. And that's what forms the structure. So this is the sequence, and it ends up folding and looking like this. So it's an alpha helix and a beta sheet, kind of, that are kind of brought together by that binding to the zinc. You could imagine that if you remove the zinc, what's to keep it like this? Maybe it would just flop like this. Okay, it stabilizes the protein structure so that it can carry out its function. Okay, who's completely lost? This does end up being a small laundry list of protein structures, but I think it's useful because if I just tell you some transcriptional regulator binds to some DNA and it activates gene expression, you understand it at this level, but if you start to think about how transcriptional regulators bind to DNA elements, you're going to understand it at this level. And so try to elevate the understanding. Okay? Thoughts? Nothing? Okay. Okay, so cysteine and histidine have properties that allow them to bind zinc, so it's intrinsic to the amino acid. Who's taking biochem? So who understands this? Because you probably had to learn all your amino acids and the biochemical properties, right? This kind of makes sense. Cysteine has this SH, so it can bind to zinc, and then histidine has this amino group which can bind to zinc. Okay. So some gene regulatory proteins have multiple zinc finger repeats, so a lot of that over and over again, and makes multiple contacts with the DNA. So here's an example of one. So 1, 2, 3 zinc ions that are going to bind in 3 different places. So that makes a very strong interaction. So it's not one helix kind of sticking in a major group, it's multiple structures kind of sticking in. Yeah?
So can you go over how the zinc is connected again? Is the cysteine and histidine part of it, or is it part of the protein?
So zinc is just a zinc ion. It's just an atom, right? And it's zinc plus plus, so it's got a charge. And the cis and his are amino acids that are part of the protein. And they have biochemical characteristics that allow them to interact with the zinc. And that's, so you can kind of imagine if you're creating, if you're building something, and it's really long and floppy, you might decide, and you want it to be like this, you might try to decide to connect it somewhere in the middle to bring it together. And so zinc's kind of acting to stabilize the structure at a different location. And so that's what you can see here. Here's your protein alpha helix of part of your protein, and here's another part of your protein, and they're connected. So right, it starts here, right here at the amino terminus, follows this way to helix, and then ends here. So you can imagine, if you didn't have this zinc ion here bound by the his and cis within the protein, you can imagine this would kind of flop back, right? There's nothing keeping it bound up this way in that structure. So it's actually part of maintaining the structure. Does that make sense? So if you even mutate one of those histidines or cysteines, 1, maybe 2, to alanine. Alanine cannot bind zinc. You'll lose the structure. And I actually, during my PhD, worked on a protein which we thought had a zinc binding domain, and that's exactly what we did. Is we mutated the individual cis and his and showed that didn't bind zinc anymore and showed that the protein lost its function. Zinc binding assays require radioactive zinc, and that was really interesting to work with because radioactive zinc is not one of your better radioactive things. I forget which zinc it is, but the guy came with the radioactive zinc, and we were like, well, do you have anything to protect us from the radiation? And he was like, oh yeah. And he brought lead bricks that were 7 inches thick. You know how heavy that is? He had to like bring like a cart to carry these bricks that were this big because zinc, like the emissions from radioactive zinc are so insane they can pass, it takes 7 inches of lead to protect you. You know, you go for an x-ray, they put that lead apron on you, that's like nothing, right? It's heavy, but it's nothing. That's 7 inches. It was so insane. So we would like do our thing and then like run out of The good thing is it didn't go very far. So like when you're working with radioactivity, you have to think about what can it pass through and how far can it travel, and each isotope is different. And so zinc, that can only go like arm's distance or something. It can't go very far. So you'd be like, no, I'm going to get inside the room until it's time for washing it. Anyways, it was no fun. Okay, I guess we're done for now. I'll start here on Friday.